I will tell you about our results of the analysis of large-scale DNA metabarcoding study on Myxomycetes. The results that I will show are very preliminary. We haven't conducted of all the analysis that we want. It's just first initial screening of the results, and we plan to extend the data that I present with other data sets as well. So, as you all know, a huge body of data on Myxomycete distribution, ecology, and diversity has accumulated in the past hundreds of years of Myxomycete research. And all this data is based on records of Myxomycete fruiting bodies. But if we look deeper into a particular species, look at its genetic structure, we often see that what we thought was one widely distributed species turns out to be a complex of multiple cryptic species with often with limited geographical distribution, as was shown, for example, for Physarum albescens with at least 18 cryptic species, or with for Didymium nivicola, with northern hemisphere populations strikingly different from the southern hemisphere populations, or with Lycogala epidendrum complex, as Dmitro has shown you 2 days ago and in his wonderful publications. So actually, what we know on Myxomycete distribution and ecology is often not at the species level, but at the level of species complexes. Another problem is that all what we know about their distribution is actually distribution of fruiting bodies and not the active stages of the life cycle. We have almost no systematic knowledge on distribution and ecology of Myxomycetes as active stage of life cycle, as Plasmodia and especially Myxamoeba. About Plasmodia, we have a little bit more records in published literature about where we can find them, and have much more observations. But about Myxamoeba, we have only quite anecdotal records. And I keep a small collection of weird sources where Myxamoebal strains were isolated from. My favorite ones are clinical samples of human feces, or coelomic cavities of sea urchins, or gills of freshwater fishes. Recently, it was also updated with water from acid rock drainage with extremely low pH and heavy contamination with metals. They still can survive there, some species. And recently, a personal communication from Alexander Kudryavtsev came, who studies amoebozoans in deep sea bottom sediments, and he isolated Myxomycetes from these sources as well. Yeah. And if we look at amoebozoa more in general, it's now considered that sporocarpy, flagellar apparatus, sacs, flat amoeboid cells, tubular amoeboid cells, and encystment are the traits that the common ancestor of all amoebozoans possessed. But during the evolution of amoebozoans, in different lineages have lost different combinations of these ancestral traits, as you can see on these examples. And these examples are based on slides from Fred Spiegel from XM 2017, just to mention it. But if we look at Myxomycetes, all species that we know possess all these stages, all these traits. But maybe it's a circular dependence. Like all species that we know from protein bodies possess these traits, but maybe those who do not possess some of these traits, we just don't know because we don't observe them. Many of you probably know about the genus Hyperamoeba, described 100 years ago, with amoeba that can form also flagellated cells and cysts, but never were observed to form plasmodia or fruiting. And later genetic studies have shown that actually the isolates of Hyperamoeba fall into various branches within the dark spore clade of Myxomycetes. So it's still unclear if these strains really can never, at any conditions, form plasmodia or fruiting, or just these conditions were not provided to them, or they lacked the compatible mating type isolates co-cultured with them. We don't know. Maybe they have indeed just lost the ability to produce these stages. So what we need to increase the resolution of our biodiversity and ecology research in Myxomycetes is a method that is not dependent on fruiting and that has high taxonomic resolution to avoid the problem of cryptic species. So one of the methods that could do this is DNA metabarcoding or amplicon metagenomics. Briefly, it's a method based on the environmental DNA extracted from various samples. And then a PCR is done to specifically amplify the DNA barcodes, some short sequence fragments that allows for taxonomic identification of the organisms in this environmental DNA sample. Then these barcode sequences obtained with high throughput sequencing technologies or next generation sequencing technologies are compared with reference sequences, sequences that have taxonomic annotation at species level or higher level. And then we can get a distribution of our taxa in the studied samples. So when we deal with metabarcoding data of Myxomycetes, there are several important questions that we want to know the answers for to be able to interpret the data properly. So one of them is, do actually trophic stages of Myxomycetes in soil show a similar biogeography to what we know from sporocarp-based studies? And also, do DNA metabarcoding data actually reflect local Myxomycete diversity, or maybe there are spores of Myxomycetes abundantly flying around all over the globe, and thus averaging, homogenizing the sequence signal that we will find in metabarcoding samples. We have compiled a data set of 18 surveys. Some of them are our unpublished data. Some of them are published studies published by us or by other research groups. And they constitute in total almost 800 soil samples. All these surveys targeted the same DNA barcode region, the V2 region of the 18S rRNA gene, with the same set of primers. So we were able to process all this data together in the same pipeline. Conveniently, these samples mostly fall into two transects. One through Europe from Spitsbergen Islands to south of Spain, another in East Asia from Kamchatka Peninsula to south of Vietnam. And we have also, so to say, an outgroup survey conducted in Costa Rica on one of, as a regional transect in one of the volcanoes. All samples were classified into floristic kingdoms and some subkingdoms according to recent publication on floristic regionalization of the world, and floristic provinces following Takhtajan publications. And they were also very roughly tentatively assigned to plant community types. The pipeline was research-based with some custom Python scripts. And as taxonomic units, we used amplicon sequence variants. These are also kind of OTUs that are probably more familiar to you, but with 0% similarity threshold. These are basically just denoised sequences where every unique sequence variant is is its own ASV and is treated as a taxonomic unit. And we used a similarity-based taxonomic assignment, first using Silva 18S dataset to filter out all non-Myxomycete sequences. And then we used a custom-made Myxomycete 18S reference database based on sequences from GenBank, from our MyXosec database, and some unpublished data. In total, almost 23 million reads were assigned to Myxomycetes, and they constituted almost 4,000 different ASVs. And only less than 16% of them we were able to assign to particular species with a 99% similarity. And total, there were 127 identified species, 26 genera from 9 families and 5 orders. Most of the identified species split into more than one ASV. Thus, these ASVs might represent intraspecific genetic variability for some of the species. In other cases, they might represent hidden diversity. Cryptic species within the same morphological species. The SV occurrences, frequency curve of SV distribution by sample, shows this quite typical for Myxomycete distribution, but it's extreme variant with just 2 ASVs appearing in more than 100 samples from 8 to 9 different study regions. And they represent this huge peak of super widespread species. And these SVs were assigned to unknown species in Stemanates genus. And another one is Lamproderma arcirioides, Niviculus species. Then 41% of all ASVs appear in only one sample. And you should know that this is already after a very stringent quality filtering and removing all singletons and all sequences that have less than 10 reads in at least one sample. So let's take a look at this non-metric multidimensional incomprehendogram. It's difficult to see any big pattern when you look at it like this. But here the phylogeographical provinces are marked by shapes of the dots, and plant community types are indicated with the color. You see even like that there are some non-random distribution of colors and shapes, but let's look at it more in detail. So here colored are only Arctic, alpine, and subalpine habitats of Eurasia. And you see that they mostly group together, occupying just roughly half of this multidimensional space. If we look at now at temperate and taiga forests of Eurasia, we also see that they tend to group together in the NMDS. Then tropical and subtropical forests form even more compact groupings in this graph. Then let's take a closer look at the tropical communities. Here, the subtropical and tropical Vietnam forests show for this elongated cluster of samples. Then subtropical China samples are also close to them in the same line. And Costa Rica samples from tropical forests in Costa Rica group closely with tropical parts of the Vietnamese transect. Now let's make an NMDS and other analysis only on the East Asian transect. Then we see even more clear this clustering of the samples coming from different parts of this latitudinal transect. And if we do a neighbor joining tree of the samples based on distances in sample communities, then we see almost perfect grouping as well of samples coming from the different parts of the transect. Here you see a differential hit tree built for all the samples together. This is not a phylogenetic tree. It's a taxonomic hierarchical tree, just illustrating the taxonomic groups that appeared in samples with dots, diameter and the thickness of line reflecting the amount of ASVs assigned to this taxon. And here you see results of the comparison of the communities of holotropical kingdom and holarctic kingdom. The colors here represent the significant differences in abundances of ASVs in either. So the green branches show the taxa that are significantly more abundant in holarctic region, while brownish branches show taxa significantly more abundant in holotropical regions. So what we see here is that, not surprisingly, tropical regions show that unknown taxa are much more abundant there than in holarctic region. It can be explained first by the simple fact that most of the sequencing data comes from not from tropical regions, but from temperate or boreal zones. And also, as Martin said in his keynote lecture in the first day, it might be that although we don't see that much fruiting in the wet tropical forests, maybe there are non-fruiting species that just exist there at trophic stage forever without ever fruiting, without ever forming fruiting bodies. Maybe because they just cannot get dry in ever moist environments, or the conditions are just always so stable and nice for them that they don't need to form fructifications, which usually happens when the conditions become not so nice for them, as Johan mentioned in his talk just before me. And if we look at the holarctic region, we see that, again, not surprisingly, the branches containing many nidicolous species of Myxomycetes or snowbank species are significantly more abundant in the holarctic region, such as this Meriderma, clades of the tree, a genus almost exclusively containing only nidicolous species. Also, Lampradermatetsy that has many nidicolous species, but also some Physaraceae and Stemanitidaceae. Then here you see the occurrence table of the nidicolous species in the studied samples, grouped by studied regions. And regions are grouped by bigger regions. So the more red is the cell, the higher percentage of samples from this region contain ASVs assigned to nidicolous Myxomycetes. So you can see here the list of 10 most abundantly occurring nidicolous species in all the regions in total. But also you see that although most of Yeah, although naviculous species are more abundant in European mountain transects and lowland boreal regions and Arctic and subarctic regions, there are still some occurrences in subtropics and tropics from both East Asia and Costa Rica. Now let's take a closer look at some of the species, just as interesting examples. Polyschismium nevadense, a species recently described from Sierra Nevada mountains in south of Spain, indeed, even in DNA metabarcoding data, occurs only in Sierra Nevada. This nicely confirms the fruiting body-based observations. Then Diderma kamchaticum, another species recently described, with a vast majority of the specimens coming from Kamchatka, and only one observation from France. But at the level of DNA metabarcoding data, we see that actually this species can be found in all European mountains that we studied with DNA metabarcoding, including the Kibin Mountains in the Kola Peninsula and Alps and German Alps, French Alps, Sierra Nevada, and the Caucasus. But it is completely absent from lowland regions and from the tropics. Another example is Diderma meyere. It was never found fruiting in lowland boreal forests, where intensive naviculous species Myxomycetes surveys were conducted for many years. And it was not found in Kibin Mountains, but it was detected in soil from these regions, from Kibin Mountains, where it never fruited, and from lowland boreal landscapes in northwest Russia. And it was never found on Kamchatka in any state, neither as fruiting bodies nor as metabarcoding sequences. Then another example that shows that there can be false negatives as well. Diderma microcarpum was found in Sierra Nevada and the Caucasus as fruiting bodies multiple times. But it is absent from the metabarcoding data from these regions. So we should be really careful with interpreting the absence information. But it's always the case for any kind of absence information. You cannot just simply prove that something is absent from somewhere. Then another interesting thing is you probably remember the Dichopsis resinae ad int species that Martin mentioned many times in his keynote lecture, which has very peculiar ecological niche in stem wounds of coniferous trees. And we have not found it in any metabarcoding data from any samples in soil. So it's not yet just another naviculous species, although it is frost tolerant, obviously, and it indeed seems to have a very specific narrow niche in these stem wounds. At least it's not detectable in soil so far. So the preliminary conclusions, it seems that assemblages of soil-inhabiting Myxomycetes indeed show pronounced clustering by geographical regions where the samples were collected and by plant community types from which samples originated. So most of the diversity signal in soil metabarcoding data of Myxomycetes indeed seems to have local origin and not just some random signal coming from spores flying everywhere. Then some species show a distribution similar to what we know from fruiting bodies, but other species show a much wider distribution to what we know from fruiting body records. And majority of the species in metabarcoding data seems to be rare, and only very few species are very widespread. And most of the soil-inhabiting species in tropical regions, especially, remain unknown. to us, and it's still not clear, do they have a fruit or not? Or maybe we just didn't sequence enough species from these regions. I would guess that it's both. Yeah, and yeah. Then I'm very grateful to all the people who participated in soil sampling and sequencing. And here you can also see published datasets that we used in our analysis. And funding for the study came mostly from the DFG project and from the project ECOLAN of the Joint Russian-Vietnamese Tropical Research and Technological Center. Thank you for your attention. Do you have any questions?
So, as you know, I'm not from the field, but I wonder, so if we find fruiting bodies somewhere, how is it possible, or what the explanations are for not finding any sequences in soil in there? So you mentioned one example in this blue reddish table, where many fruiting body records are there for certain regions, but you do not find them represented in your environmental DNA.
You mean how can it be explained?
Yes. Yes.
I think the reason is the high variability of soil as a substrate. Like at the micro niche level, it's extremely heterogeneous habitats, and if you have like a small depression, it's one microhabitat condition. If it's just a little bit elevated or shaded or in sun, it all differs a lot for the Myxamoeba living there, I guess. So I think the distribution of species in a particular region is extremely patchy. So wherever they find appropriate microhabitats, they might be abundant, but if you sample 1 meter to the left or to the right, you will not find these species. So yeah, our samples are always like tiny fraction actually of the study territories. So we have to expect lots of false negatives.
Yeah, I had also a similar question if you sampled soil from the same localities as fruit bodies, but you already answered that not. And the second question is how can you explain the presence of Niviculus Myxomycetes in other forests where where there's no environment optimal for them? They are only single records, yes, if I understood well, but
not always single. Sometimes they appear in several different samples, in several independent samples from some subtropical or tropical region, or just or even a mountain region where it could, in theory, fruit, but it never was observed fruiting. So we don't know. Maybe the niche of actually Myxamoeba of these niviculous species are much more widespread, and then they can persist not only in niviculous conditions, not only under thick snow cover, but also in just in regular soil conditions in other regions. But maybe they do not get conditions for fruiting there or for accumulating large biomass. Yeah, it's an interesting question. Yeah, we don't have an answer for that. Yeah.
So don't you think that it can be also the long distance dispersal of spores, so spore deposit from a long distance from fruit bodies, and you just detect only the presence of spores?
It might be an explanation as well. Yeah, although there are too many unanswered questions. We do not know how far is spore fallout from really distant regions. We do not know how long do the spores persist in soil, how fast they are degraded by microorganisms and fungi. Also, we don't know if the spore is degraded and the content of the spore is released and partially degraded, how long is this extracellular DNA preserved in soil. It's known that extracellular DNA can stay stabilized for centuries and 1,000 years. That's how all these ancient DNA studies are conducted. They target this extracellular DNA. But mostly it should be fragmented into really very short fragments. And our DNA barcode region is around 350 base pairs, plus minus. And at least what I think, probably it should not be able to amplify the old extracellular DNA. I'm not completely sure of it, but that's just how I think for now.